As I mentioned earlier, it's such a great opportunity and an honor to be able to talk to everyone at FSU, people who might be joining from other organizations. And I am just really missing the opportunity to talk to people about science and what it's like to be in science. So as is part of many academic talks, I think it's important to say that my views don't represent the University Medical Center, but they also don't represent any of the other biologists who might know that they're, who wish they were better chemists, the entire state of Florida, any of the other Filipinx people who love making lumpia like me, or the many, many other scientists who also like to make and create art. So my views essentially, although I do talk about many different kinds of people and many different fields, it's important to note that my views, as for many other people, represent my experiences, my heritage, and very importantly, the village of people who've supported me. I think science is often advertised as this isolated event, and much of it is isolated, sitting in labs, working by yourself, and the kind of gambit of trying to defend your work to other people. But I think that one of the main points I want to make is that in order to, really pursue what you want in STEM, you really need to find mentors and to be a mentor. So I'm going to emphasize that as much as possible in this talk, finding mentors in your classes, in the lab, at choir practice, and being the person that you wish was there for you when you were first joining STEM or really any pursuit in your life. But before we start talking more about me, let's talk about you. Please join this survey. You can join on the QR code here, or using your phone, you can go to this website or text this code to 22333 to join. So I'll give everyone about 30 seconds to join on so that we can then go through and find out who is here with me today. Isn't technology cool? I don't know if they still do this at Florida State, but they used to have like eye clickers where you had to like come into class and have these like ABCD buttons. And if you forgot yours, which I lost like 3 of them when I was in college, you basically couldn't answer any questions during class. All right, so I hope everyone is in. And now I think what's really helpful for me is to kind of find out what is your age? So basically what I'm looking for is, are you in high school? Are you in grad school? Are you already working in STEM? Because I don't want to necessarily be preaching to the choir, but I also want to make sure that if we have people in younger groups, that I'm touching on information that I didn't have access to at different stages. So it looks like we have a decent number of people who are probably in college, a lot of people who are now, probably well established in their careers. Let's see. Awesome, yeah, so it's great to see that we have about 30% of people who are still working on figuring things out. I mean, we're also trying to figure things out, but yeah. Next, are you currently involved in STEM, or are you curious about getting involved in STEM? Pretty diverse group of people. Majoring in STEM, great. Cool, so no one who is not in STEM so far, well, in terms of college. Very cool. All right, next and most importantly, what is your favorite holiday and why is it Halloween, obviously? Getting a solid representation for Halloween, very proud. Christmas is pretty solid too. Nightmare Before Christmas, good Halloween, Christmas mashup. All right, yes, excellent. Christmas or Halloween for the win. All right, how does STEM make you feel in one word? This can be a pretty wide range of responses. It's helpful to note that if you see an answer that you agree with, if you submit the same answer, it will make that word bigger. So we'll kind of get an idea of the proportion of feelings for STEM. These are really great responses. Busy, for sure. Eureka, curious. Curious is probably the best thing you could be. Honestly, the way that you're most resilient is to take all of that crazy negative data and just be more curious about it. Busy, yeah, incredibly busy. Thanks all for your responses. All right, share your future goals. So this is another one that has an extra level of interactivity. If you see something that you align with, then you can upvote or downvote, so we can also get an idea of what people are interested in. So we can see if we have more people who are interested in going to grad school. Very cool. There's something very special about working with students who are interested in STEM. Nice to see people considering grad school. Hopefully this talk will help someone with that. All right, well, thanks everyone for your answers. This really helps illuminate things for me. I will process your data promptly. But yeah, so welcome again, everyone, to this talk. And the title of my talk is How to Become a Scientist in 100 Quick and Easy Steps. And the reason why that's the title of my talk is because becoming a scientist is one of the hardest, most complicated, and riskiest things you can ever do. And I think, for some people who might not be acquainted with the process, it can be risky because it can be very dangerous, exposing yourself to things in the lab, mentally and physically exhausting. It's pretty risky because for a lot of things like grad school, your entire life is tied to the opinion of one person. And another thing that people don't appreciate is that it's very financially risky, where you experience a delay in legitimate pay for up to 10 years. And I'll talk a little bit more about that later. But the structure of my talk today is a little bit about my journey so far. Obviously, this doesn't represent other people's experiences, but for me, as someone navigating college and grad school, it would have been really helpful to hear more specific examples about what type of research people did, how they went through school, and how they pursued their future career goals. So I'll emphasize again, one of the reasons why I'm talking about this is because I want people to see how many people helped me along the way. Yeah, so this is my kind of chronology. I graduated in high school in 2010. I graduated from Florida State in 2013 with my bachelor's in biology. I went to grad school at UAB in Birmingham, and then I did a 3-year postdoc training up until this past March. For example, this is helpful to note because when I was a postdoc, or when I was a grad student, I didn't even really understand what a postdoc was. So a postdoc is basically you have your PhD, you've shown that you can publish papers and that you can write grants. And this is basically your first opportunity to really stretch your legs and start developing really independent research programs. And so a lot of people will go to grad school and then do 1, 2, or 3 even postdocs before they then start looking for faculty positions. But what kind of put me in this direction in the first place was my parents. My dad is on the far left. His name is Carlito. He's a respiratory therapist. So he's someone at the Veterans Affairs Hospital who actually puts patients on ventilators. And so this is him. This is a recent picture of him in the COVID unit getting suited up to treat patients. My mom has been a nurse for many years. She's in the middle. Her name's Isenia. She has worked in the ICU, in the trauma clinic, in the ER. She's worked in trauma with traumatic brain injury patients. So she's done basically every form of hardcore medicine as you can imagine. And so when I was in middle school, I was actually thinking about becoming a pharmacist because I wanted to be involved in healthcare, but I also, was really wary of providing clinical care because it was hard and scary. I'll talk a little bit about how I ended up going to research instead. But basically all that to say is my life up until now has always been on this trajectory of patient care and how to, care for people at their worst moments. And so when I went to college, I didn't really know what I wanted to do. I knew that I wanted to study biology. I didn't know how to get involved in biomedical research yet. So I looked at a variety of different labs and ended up joining a lab that studied really basic fundamental genetics. And so instead of studying flies or instead of studying like rats or humans, we use Drosophila, so fruit flies, because they grow really quickly and it's very easy to manipulate their genetics. So we were able to introduce a variety of genetic variants or mutations. And it's kind of a typical strategy for biologists wherein in order to figure out what something does, you have to break it. So you take gene A and introduce mutation B, and then see how that changed the morphology or the functional or structural elements of an organism. So for example, we used a form of microscopy called confocal microscopy, and we would dissect out the ovaries from these flies. Their ovaries are actually bigger than their heads. And then look at different stages of egg development to understand what different genes are involved in terms of the formation of these eggs. But also things in general like cell-to-cell competition. So I studied that and I said, okay, like now I've got a good handle of genetics. Now how do I apply that to healthcare? So I applied to a few graduate programs and got a fellowship at the University of Alabama at Birmingham. I received a fellowship in translational research. So there's a wide variety of different programs. This is another thing I didn't know in college, was like, I didn't know that there were these different programs, that there were these options. All I knew was, okay, major in biology. And then I could have gone into any of these programs. I was pretty undifferentiated. I didn't really have any opinions about these things, but I did know that I wanted to do something that had to do with actual therapeutics. So I applied to the pathobiology and molecular medicine program. And so within that program, what's also great about these kinds of programs is that you don't have to be married to one program or one field of research and one lab for the rest of your life. So a lot of programs like those abroad, in different countries, you have to get your master's degree first, and then you have to secure funding. And then you're basically stuck in that one lab for 5 to 7 years and you don't have a choice. But at UAB, I was able to do 4 rotations. My first rotation during the summer was actually at a local research institute outside the university, where I studied the differentiation of glioblastoma or cells or cancerous cells within the brain. I worked in another lab in the pathology department where I studied stem cells and how they can switch from being kind of fatty cells to ones that help form bone. I worked in another lab where this lab was pretty interesting because the main PI, so I'll say principal investigator, is also the mentor, is also the lead scientist for a lab. He was able to collect samples from actual patients and then bring them to the lab, and then we could work with them. So it was highly translatable, which is how we describe research that goes from a basic bench in a lab, moving it towards the bedside, so in the actual hospital with patients. But I ultimately ended up in joining Eric Sorscher's lab. This is my mentor and I at graduation, far right, studying cystic fibrosis. So a lot of people go into research, because they know someone who has breast cancer, Alzheimer's disease. I didn't know anyone with cystic fibrosis when I went to grad school. I was just really interested in, okay, what's something that I can help study genetics and medicine at the same time? And this seems like the perfect example. And at the end of the day, I actually picked his lab not because it was the perfect scientific fit, but because, we vibed on, the way we saw the world, kind of the way we talked to people, the way that we, treated people. And I think I tell a lot of students nowadays is, instead of being married to the scientific element that brought you to school in the first place, you really need to find a mentor um, who can support you as a person. Someone told me, this is like a really, kind of blunt and maybe bizarre way to put this, but they said, when it comes to grad school, picking your mentor can be more important than who you pick to get married, because if you pick the wrong mentor, you might get divorced because of how much control or perceived control your mentor can have over your life when you're in grad school. So I'm very grateful that I joined his lab, and I definitely wouldn't be here without his advocacy, both in the lab and also in life in general. Yes, cystic fibrosis is, to this day been the biggest example of can we study someone's genetics and determine how sick are they going to get and how do you pick the best treatment for them. And so cystic fibrosis is one of the most common fatal genetic diseases in the United States. It affects over 200,000 people. And it's really, it's a disease that affects basically any part of your body that secretes mucus. So any part of your body that needs regular lubrication. And so your lungs can become thick with mucus that causes an increase in the amount of bacteria that grow. You have abnormal sweat. These patients often also have insufficiencies in their pancreatic function. And all of this occurs because a patient will have one mutation in one gene in their body. And this, the fact that there is this loss of proper lubrication is indicative of the fact that these mutations occur in a part of the cell that regulate water flow. And so this protein or this part of the cell is called CFTR. And so it's basically a chloride channel. And so the way that the chloride ions move in and out of the cell affect where the water ends up going. So that's why the mucus becomes thick in these patients. And there are a variety of ways in which you might lose this transport of water. You can, there are many different mutations that can modify whether or not that chloride is able to move. And now so now you can see that the chloride is trapped within the cell. And that mucus becomes contaminated with bacteria. So as was shown on the slide, there are actually over 1,600 different mutations that cause cystic fibrosis. So an incredibly insane amount. And what's been really difficult with studying cystic fibrosis is that many of the research programs have targeted one specific mutation, this delta F508 mutation. And so the gist of our program is that there are rare patients who have mutations that might be better candidates for treatment. So for example, on the far right, wild type is basically someone who doesn't have cystic fibrosis. Delta F is someone with severe cystic fibrosis, and P67L is a mutation in patients with milder disease. And you can see that with treatment for P67L, this black band is similar to healthy patients. So you have almost complete countering of that disease, versus with delta F508, you see almost no change, even though the drug was designed for it. So the situation you end up in is some patients all of a sudden can contemplate a longer lifespan from, late teens to now late adulthood, but some patients don't qualify for it. And so what we were basically doing was developing tools in the lab that would predict how well patients responded to different drugs. So you can see here again, this delta F508, at baseline, you have 0 function. And then with the drug, you have, about 8, moves from 0 to 8. And then for P67L, you go from below 50 to over 150. And so we were basically saying, like, there aren't enough patients to do a clinical trial, but we're able to show that we can predict in the lab using cells that these patients will respond well to the drug. And we're also able to do some molecular modeling with collaborators, as shown on the right, to show where these drugs bind, and hopefully that will tell us whether or not other patients will respond well to these drugs. And so as a postdoc, I went from studying one mutation to studying two more. That was basically, there's 1,600 mutations. How do we expand these tools to other things? And on the right, you can see now we have mutations such as G85E that don't respond to the drug, another mutation that does respond well. And so it was after this time that I graduated and was thinking, okay, what do I do with my training? How do I, what is a new tool that I can develop that will help ask similar questions to what I did in grad school? So then I joined the Plata lab at Vanderbilt in 2018, and I learned how to use this fancy box next to me, the bottom left, and it's known as a mass spectrometer, and it basically helps you take things within a cell, break them down to their component parts, and actually identify what is inside of the cell. Instead of, what I showed you before, those blots where it's like one protein at a time, now you could look at thousands of proteins at once. And so the middle here is a scheme of how we're able to pull down one protein out of the cell and see what's attached to it. And basically, on the right, you have a heat map where you can see wild type is this gray baseline. L206W, you see that at baseline, there's a lot of red, and then it changes to be similar to wild type with the drug. And then for more resistant mutations, you see no change. And so this helps us identify things in the cell like involved in translation or protein folding. What are the areas that need to be targeted in order for these drugs to work well? Kind of fast forward to 2021 in March. Now, instead of studying one disease, I study a variety of diseases. I work with 4 different labs. And so now I study chronic bronchiolitis, which can be caused from smoking. We specifically study a cohort of patients who are vets from Iraq and Afghanistan who've been exposed to sulfur dioxide, as well as interstitial lung diseases and idiopathic pulmonary fibrosis. These can be caused kind of having unknown origins or via some type of genetic mutation. So you can see how, although I've gone through many different iterations of different kinds of research, I've been able to follow a cohesive kind of line of questions throughout my career. But in reality, this is the story I usually tell people, college to right now. But in reality, the thing that I often find myself leaving out because I'm scared of people judging me is that I went to nerd camp. And, there was always this feeling that I would be targeted for having an unfair advantage, even though programs like this that were targeted towards minorities were meant to help to counteract the disadvantages that come with being from an underrepresented background in STEM. And we live in this world where we're increasingly finding the cracks that people fall through and trying to create these programs to better support people in STEM, but then also shaming people for being diversity hires or tokens. And so programs like this, I was in it, from about 7th, 9th, or 10th grade, and they show you basic lab techniques, and you work in a lab for the first time before you even ever going to college. And then it's about a 10-year program, and people down the line can actually work for like actual industrial labs or work at the NIH. And so it's been incredibly useful and has changed, hundreds, maybe even over 1,000 different students. And I can say that if I didn't have the specific training that was designed for children from historically excluded backgrounds, I would be prescribing treatments instead of developing them. And obviously, for a lot of people in STEM, becoming a nurse or a doctor or an engineer is awesome. And if that's what you want to do, you should do it. But you should do it because you want to, not because you didn't realize that there were other opportunities out there for you. And so, without programs like these, I wouldn't have known that becoming a scientist was even a choice. And so, I'll talk a bit more about what it's been like, navigating my identity and, the cultural entities that represent who I am. But we usually think of diversity as one thing. And so like in another example of considering diversity, especially within research and healthcare, is something that I was working on recently. As I mentioned, we worked with some soldiers, and we were planning a clinical trial for soldiers who were exposed to these toxic gas fumes abroad. And we were forming the board of people who would be responsible for designing the study and organizing patients. And at one point I realized, like, hey, shouldn't we have an actual soldier as part of this board of people who are deciding how they should be treated? And this is something that is overlooked all the time for clinical trials and many different things. And, you know, as I mentioned, out of my fear of being judged and saying like, oh, like you were only able to do that because you received XYZ extra treatment, I completely avoided any type of organization that, you know, was dedicated to women. And it wasn't until my lab moved to a different city and I didn't know anybody, and someone reached out and was like, hey, like we have this organization for women in science, that I started working with them, and it just completely changes how you see and work. I was also really lucky at the time that my best friend in grad school, shown here, Lindsay Smith, who's now a PhD, her mentor was the dean, Laurie McMahon. And my mentors have always been white men who've always been super supportive and like wonderful people. But there are many things that happen to me in my life and to other women of color that you just can't talk to your boss about unless they've had those lived experiences. And I definitely wouldn't have been able to survive some of the things that I experienced in grad school without advice from a woman. And I also was able to organize a conference with these other two women in Atlanta. And it just completely changes not only how you navigate the world, but the kinds of scientific questions that you think of. Lindsay here, she studied Alzheimer's disease in terms of the effects of estrogen, because a lot of studies that they do in animals, these are animals that are premenopausal. And so they still have a lot of estrogen, but for the onset of Alzheimer's disease, you're working with women who are postmenopausal. And it wasn't only until the past few years that the National Institutes of Health, not required, lightly started suggesting if you're going to do biological studies, you have to include female animals. So if you ever wonder, you know, for a lot of women, if you have really bad side effects to drugs, that's because almost all drugs were developed for white men. So on that note, so my family's from the Philippines. My parents immigrated with my brothers, and I was born in Florida. And here we are with my grandma in the Philippines, with my parents. And it has been such a wonderful opportunity to come from the Philippines. And I think what's really powerful about people from the Philippines is that so many of them have been able to come to the United States because of our interest and investment in healthcare. So in the 1960s, there was a nursing shortage, and that became kind of one of the major pipelines for people to immigrate to the United States. And there's, you know, a long-term legacy for this. You know, children have been continuously encouraged to pursue healthcare-related careers such as nursing or going to medical school because it seems financially stable. Well, also many other reasons, but one of the main reasons is because it's a, you know, you're going to get a job, you know, you're going to make money, be able to support your family, and be able to send money back to the Philippines. And so this is from an interview featuring some other students from California. And, you know, they've noted, as I've touched briefly upon earlier, that only people who have the capacity to have low income for a decade, because you go to college, you go to grad school, you're a trainee, you do your postdoc, you're a trainee, that can be on average 7 years of being paid as a trainee, not as an employee, no retirement benefits, oftentimes no dental, for, you know, until you're in your mid 30s. So only people who aren't worrying about paying for an entire family or people in a different country can really make that risk. And a lot of people who do this pipeline like don't even have a decent job at the end. And so, you know, navigating this, let's see. I had never really met many Filipino scientists. I'd never met a Filipino scientist until I went to grad school. I went to a talk and someone came up to me and looked at my name and said, "Where are you from?" And I said, "I'm from Florida." And they said, "Where are you really from?" And I said, my parents are from the Philippines. And they said, "Oh, hey, like there's this new professor who just joined the university and she's Filipino, she's studying cystic fibrosis as well." So I had lunch with her, and it was this really weird thing to just like shove people together just because of something like that. But it was huge for me to see someone who looked like me actually succeed and be in the sciences. And she was a nurse, like had her RN, and then decided to go back to school and get her PhD. And then so more like 4 years went past, still hadn't met any other Filipinos. And then all of a sudden, when I moved to Vanderbilt, I met like 12 Filipinos who are all scientists. And now there are organizations like this, Association of Filipino Scientists in America. And the past few years, I've discovered there's so many new resources for people to kind of engage with people with similar backgrounds. This is 2019. It wasn't around when I was in grad school. And so, your perspective on what's possible is just, fundamentally changed. And having this in mind is really important because culturally, there's still a lot of backlash for many different communities, but also specifically Asians in terms of, what we're allowed to do. Even though things are improving slowly, most faculty, the demographics of most faculty do not represent the diverse populations that actually attend these universities. And so when I was in grad school, I went to a talk, or I gave a talk in grad school, and one of my, role models, someone whose research I followed throughout all of grad school, whose research I really respected, who I like fangirled over, came up to me and gave me a hug. And I was like, "Oh my gosh, like what is happening? I don't understand." And he said, like, "You did a great job," but also like, he was like really overwhelmed and just said, "It's so powerful to see someone like you, like us, up there giving this talk at this conference." And he said, "I tried to become a professor, and I went through all the different series of interviews, and the lead chair of the committee that was deciding whether or not he would be offered a faculty position told him that Asians aren't supposed to be professors, that Asians are meant to work in the lab. If you look at many labs, like there are a lot of staff scientists, a lot of research associates, a lot of postdocs who are Asian. So you're supposed to be doing experiments and doing what you're told. You're not supposed to be a professor." So, this is a thing that's still very much alive and well. Now he's like a really successful professor, has had his own lab for like 20 years. So he's able to push back through that. But the ways that we think about, underrepresented minorities definitely mold the things that are possible. So what we often use, the URM, actually implies that there's a deficit among these groups rather than the lack of representation being a byproduct of perpetuating these work climates that limit people's opportunities. Like I mentioned, financially, in terms of stereotypes, and also like cultural expectations from groups such as your own families. Yeah, but now there are many different ways to find your own communities. Going on Instagram or Twitter is really powerful in terms of looking at different hashtags and identifying specific scientists that you might be able to follow. One woman that I follow very closely is Corey Grayson. She's huge in the STEM diversity field. She has these 3 handles, as you can see on the bottom right. So I highly recommend following her. A good friend of mine, Stephanie Castillo, she was originally a PhD student in the chemistry department. And now she invented her own PhD program for science communication. She has a startup company called Future Doctors, where she interviews people in STEM from diverse backgrounds and talking about what do you want to do, how did you get there, and how do we support other people. And so that brings me to the last part of my talk in terms of diversity in healthcare. And so, as noted by this clinician here, being a minority in our social context will literally destroy your health on a molecular level. And the way that we usually contextualize race in medicine is that people via genetics are automatically inferior because of their genetics, not because of the socioeconomic context that they've been forced into. And so, for clinical trials, and for very good reasons, people of color are resistant or, terrified to participate in clinical trials. If you look up, the Tuskegee trials and the ways in which these soldiers who were afflicted with syphilis were told that they were being treated when they actually weren't, because they were supposed to be this long-term, like 30-year experiment. And, to this day, for many studies, for example, like as an Asian, Asians basically are completely non-represented in clinical trials, even though we have completely different metabolisms, which means that you can take a certain drug, and because your body doesn't fully metabolize it, you end up with twice as much of it in your bloodstream, which you can imagine can have devastating effects on your body. And of course, as I mentioned before, the lack of representation in terms of actual physicians and researchers. But it's much more than just clinical trials and the people who run them. Racist normalization algorithms are deeply embedded in healthcare. If you walk into the hospital tomorrow, there are literally buttons and algorithms that just switch between white and black and sometimes Hispanic, never Asian. And so one example is for measuring kidney function. There's this crazy idea that if a Black person has more EGFR or like more of a certain protein in their blood, so this is basically something that normally your kidneys filters out. And if your kidneys are failing, then more of it appears in the blood. But there's this idea that if you're Black, you automatically have more muscle mass, which means it's normal for you to have more of this protein in your blood. So basically what that means is that for a 65-year-old white man, if they have an EGFR of 18, that means that their kidneys are failing and they can get a kidney transplant. In contrast, for a Black patient, if they have an EGFR that's even higher, they'll say that's normal for Black people because they have more muscle, and so they're not eligible for transplant. And so this is something that Vanderbilt has actually been working on since 2018. And they've actually been able to organize a group of medical students, not even like people who are high up in power, who've been able to put together a toolkit to say, let's stop using this race factor. And they've started implementing it on a national level. Another really insidious example, some people have seen in the media when it comes to the NFL, there were these race algorithms that basically said Black people are automatically, have lower cognitive function. So if they have traumatic brain injury and they don't perform well on these cognitive tests, that's normal. And that means that they don't qualify for any type of extra healthcare or any type of financial support, even though they're experiencing these incredibly debilitating effects of brain injury. And, that's something that I think there are some lawsuits moving forward, but there definitely needs to be more work regarding that. But one example that I've been able to interact with personally is the race factors involved in lung function. And so there's this great book by Lundy Braun that does an overview of this. But basically, if you go to the doctor, and there are these instruments that you're able to blow in, and it measures your lung capacity, so how much air you're able to hold within your lungs. And what's really bizarre is that, some of the early, these are called spirometers, when they first started characterizing what's normal lung function, there are early documentations that this work is still based off of by Thomas Jefferson. who justified racism by saying Black people have lower lung function, have smaller lungs than white people, but if you force them to work in the fields, all of that physical exertion will make their lungs bigger. And to this day, most hospitals, if you're Black, they will use a race algorithm to adjust your lung function. And it's similar to the kidney function work, where this means that they all of a sudden are not eligible for a lung transplant, even though their lungs are failing. And so one group has recently been able to take some data and show that if you use the race algorithm, a majority of the patients are considered to have mild to severe disease, shown in this brown. But if you remove the race factor, you can actually see that a larger proportion of those patients actually have very severe disease. So all that to say is that you have many different patients who are dying without lung transplant because of these race factors. And so my first grant in my current position, I'm collaborating with a clinician. We're looking at the race factor among patients with cystic fibrosis, particularly children, because many of them have to go through lung transplant, before they're even 18. And so all that to say, these are things that are deeply embedded in our healthcare system. And I think a lot of those things are only recently coming to light. But I've talked about a lot today. And, I think there are three main takeaways that, if nothing else, I'd hope that you would remember. For my story, science is really complicated and there's so many things in terms of applying for schools and things like that. But the main thing I think that will really get you through at any level within STEM is finding mentors and being the mentor that you needed when you were younger, at whatever stage. Acknowledging and embracing and using what makes you different, helping people with those different backgrounds, whether it be, your cultural heritage, or, if you're someone who suffers with depression, and if you come from a certain disenfranchised group. And also fighting institutionalized prejudices, identifying, things within your organization that you might be able to have an effect on. But yeah, thanks everyone for attending. You can go ahead and use the same website and text function to submit any questions and comments. And I'm just really grateful for the opportunity to talk to y'all.